All right. Thanks, everybody, for coming, and welcome to today's 5 Points lecture. I'm Michael Zodi, the Director of Computational Biology here at New York Genome Center. On behalf of the Genome Center, I'd like to welcome everybody here. It's my great pleasure to introduce today's speaker, Dr. Ben Voight. Ben got his PhD at the University of Chicago, working with Jonathan Pritchard and Nancy Cox, where his work focused on studies of natural selection and demographic inference in human populations. After that, he did a postdoc at Mass General Hospital and the Broad Institute with David Altshuler and Mark Daly, working on common genetic variation associated with complex disease and the framework of Mendelian randomization to perform causal inference using human genetics data. He's currently at the University of Pennsylvania as an associate professor in the Department of Systems Pharmacology and Translational Therapeutics and the Department of Genetics, and serves as the vice chair for the Genomics and Computational Biology program. In 2012, he was selected as an Alfred P. Sloan Foundation Fellow, and he recently received a Presidential Early Career Award for scientists and engineers. His research focuses on population genetics and computational biology and the impact of genetics on complex disease, with the goal of developing genetic observations into targetable mechanisms, evolutionary insights, and new candidates for therapeutics. And his work focuses on using large-scale genomics data to discern evolutionary history and genetic basis of complex disease, which leads nicely into his talk today. Welcome, Ben.
Thanks, Mike. How's my volume? Good, not too loud? Rad. Okay, so thanks, Mike, for that introduction. Very generous. Thanks for the opportunity to come share a bit of my work from my lab. I am a human geneticist, and I can't say and emphasize what an exciting time it is to live in a time where we have unprecedented access to human genomes, the ability to survey in large numbers of individuals DNA sequence variation, and then be able to take that variation, relate it to various complex diseases, as well as use the patterns of genetic variation that we find to make basic biological insights about populations and human populations in general. So what I'm not going to talk about today is any of our population genetics work on mutation rate models. It's a little blanched out. Nor am I going to talk about our work sequencing spider genomes. If you want to buy me a drink at the bar, I'm happy to talk your ears off about those efforts, maybe after the fact. But today, I'm going to really focus on our efforts thinking about human disease, genetics, biology, how we interpret specifically complex trait variation, how we can generate advantage from human genetics data. And really, as sort of, I'm in a department of pharmacology, but I'm a geneticist, so you might think that's a little strange. But really, it's the goal of which is to bias how we pursue therapeutics with human genetics insight. And if we can do that, even increasing the rate by twofold or threefold, that would have a huge impact on our ability to make effective clinical changes in practice. Okay, so I've told you that this is a great time to be a human geneticist, to think about DNA sequence variation, to relate that variation to disease. But how do we make those insights? Okay, so I'll just, for the sake of argument, I'll present to you sort of two models. You can tell me what you think about them. So one I'll propose to you is sort of like the grid search model, where you've got a large number of things you can test, and you have enough resources and time and effort and people, really, to put an individual person on each one of those activities. This will guarantee you biological discovery and insight, and if all of us had all these resources, we would absolutely do this grid search process. That is not my lab. Okay, my group is a little more agile, roguish, if you will, in terms of the number of size and the amount of resources we have. And so the implication of that is that we have to be creative and clever about what bets we place on what particular aspects of genes and biology we discover. So it means that every decision we make has to count. We can't just choose things randomly or willy-nilly. It has to be based on strong statistical support. It has to be really biologically and physiologically compelling. It's got to give us a clear hypothesis that we can test very directly. But it has to be non-obvious, because if it were obvious, our friends and colleagues at various other places would have made those discoveries well ahead of us in the first place. So the hope that I would like to leave you all with, if I leave you with nothing, is a key idea that is that computational and statistical approaches can be that tool that helps us narrow down on interesting aspects of biology that we can focus on. So what I'd like to do today is talk with you, give you sort of like 5 points-ish on some of those ideas about how we think about this particular problem. So I'll start a little bit with some work that we've continued on with trying to use human genetics data to make causal inference, taking human genetics data not just on a single trait, but multiple traits at a time to find key things that are interesting to follow up, clever tricks that we can use algorithmically, taking advantage of machine learning techniques to try to take specific sites that we think are interesting for type 2 diabetes and maybe platelet-related traits. Again, trying to identify key biological nodes that we can interrogate further. And then I'll close, if I've got some time, with just sort of like my 10-cent philosophical point about things that I think are of interest given the sort of changes that are coming up in human genomics in now and in the future. Okay, so causal inference. Really what this sort of set of questions revolve around are the central one, which is, what trait should I be spending my time studying? Okay, so what do I mean by that? Okay, so diabetes is an endpoint, right, that we're clinically interested in, but there are definitely intermediate factors that cause increased risk of diabetes. Obesity is one of them. Different lipid concentrations are another one. The same thing with heart disease. Cholesterol is definitely a causal risk factor for heart disease. Elevated systolic blood pressure, diastolic blood pressure, causal risk factor for diabetes. We know that there are causal risk factors for diabetes and heart disease. Why? Because we have great drugs that lower LDL cholesterol and lower heart disease risk. We have great drugs that lower systolic blood pressure and lower heart disease risk. So what we should do is try to identify traits that really matter that are in this causal pathway. Okay, so let me try and set up this design for you to try to give you an intuition about how this approach works. Statistically speaking, thinking about just the clinical trial setup. And the clinical trial setup at a very, very high level to a first approximation goes like this. Let's imagine we have a hypothesis and a particular drug intervention we suspect lowers. We'd like to test the hypothesis that lowers a disease risk. So what would we do? We'd collect a large number of individuals, randomize them into 2 separate groups, one group that gets an intervention, one group that doesn't or gets standard of care, measure a biomarker of interest that we think the intervention perturbs, and then ask the question, do individuals or subjects that receive the perturbation have differences, lower rates of the endpoint that we're trying to prevent relative to those that get standard of care or controls, right? So if this is true, and we do demonstrate this truly, we find 2 things. 1, that the biomarker of interest must be causal, and 2, we have a drug intervention that probably works as efficacious. So if we had this, this would be great. In many cases, we don't know what the biomarker that's causal is, and we certainly don't always have a drug intervention that is efficacious that we can test. So that's where human genetics comes in. So in an analogous way, we can use human genetic variation to test specific hypotheses about whether or not a biomarker causally relates to an endpoint. And the way you can think about this is that the genotype serves as the drug intervention itself. So if I found a genetic site in the genome that I knew carriers of that genotype had a substantial and significant difference in the biomarker of interest, then that gives me a tool, an instrument by which I can test the hypothesis that change in that biomarker lowers or changes the rate of the endpoint of interest. So this design is the Mendelian randomization design. We know about genetic risk factors. We can test that hypothesis directly. And here's like one example, in case that intuition doesn't follow you, that I think a lot of people use to sort of support this idea. And this involves loss of function mutations at this gene called PCSK9. So we know from human genetics data that loss of function mutations in this gene substantially reduce LDL cholesterol on the order of 21 milligrams per deciliter, so a substantial change in LDL cholesterol. Based on epidemiological data, we know that lowering LDL cholesterol by that amount should lower hazard of heart disease by about a quarter. So the question you ask is, does that genetic variant, do loss of function mutations in PCSK9 lower heart disease risk? The answer is yes. Those lucky dogs not only have lower cholesterol, they are protected from heart disease risk. Not only that, the degree to which they're protected closely matches the epidemiological expectation. So this gives you sort of an idea that if we could just find more instances like PCSK9 variants, more loss of function mutations that disrupt the biomarker, we can test the hypothesis that the biomarker relates to the endpoint. So again, when I ask what traits should we study, the traits we should study are examples where the human genetics data support causal hypotheses between the biomarker and the endpoint. I want to figure out not only what other biomarker, what other genetic variants perturb the biomarker, but specifically the genes that perturb that biomarker might be the next stages of which we can develop therapeutics around. Okay, so let me give you a couple of examples of cases that we've pursued in the last couple of years to give you an idea of how we can use this information both positively and negatively. So one story we published a couple of years ago involved serum urate levels. So serum urate turns out to be a causal risk factor for gout. So individuals who have gout can have their gout treated by having therapeutics that lower their serum urate levels. That is a known and established treatment for the disease, but it's been outstanding whether or not elevated levels of serum urate actually are causal for other cardiometabolic diseases, because people who have gout tend to have comorbidities of diabetes, heart disease, and cardiovascular complications. Serum urate levels turn out to be a genetically modifiable trait, so we have a vector of human genetic associations that relate exclusively to serum urate levels. That allows us to ask the question, do these levels of serum urate also alter risk to one or more of these cardiometabolic traits? So you can, if I focus your attention on the sort of top panel here, this sort of gives the bottom line for it, and the answer is yes, genetically elevated serum urate levels definitely increase risk of gout, but have no impact on type 2 diabetes risk, coronary heart disease risk, stroke, or heart failure. So, man, I'm so glad I know this now. I do not now want to do a clinical trial for lowering serum urate therapeutics on any one of these endpoints because the genetics would predict these would fail. Why is that interesting? The model systems work that's been done on this goes in the opposite direction. So you can feed mice who are predisposed to metabolic disease serum urine therapeutics, and they seem to have remediation of these endpoints. So the human genetics here tells a different story than what you learned from mouse data, but I'd be very skeptical about I'd need more data in order to convince myself that this would be a useful clinical trial to engage in. Another story that we told was about systolic blood pressure. This is a little bit more complicated, but again, systolic blood pressure is a causal risk factor for heart disease. We have systolic blood pressure lowering medication, antihypertensive. Those are great treatments for heart disease. But we also know that hypertension is a risk factor for type 2 diabetes. It's definitely correlated with the endpoint disease, but we don't know whether or not that is correlated or causal. However, again, we do know genetic variants that exclusively change systolic blood pressure rates. So now we can ask the question, does genetically elevated systolic blood pressure increase with type 2 diabetes? And the answer appears to be yes. For every 1 millimeter per mercury change in blood pressure increases risk of type 2 diabetes by about 1 to 2%. Now, this is genetic information, one piece of evidence in the causal inference puzzle. There's obviously a lot of confounders that one need to think about with respect to this. For example, the drug medication people are on, the cardiovascular complications that might be increased risk in diabetics. But this sort of gives you a clue, something that you might want to spend a little more time thinking about, because if it turned out that just lowering systolic blood pressure actually reduced diabetes risk, that would have an immediately impactful translational impact. Of course, we need a little bit more data to sort of show that formally. Okay, so just these two traits are things that we've thought about, but you can imagine as we attach genetic information to phenotypes more extensively through EHR, but also through directed efforts, you can immediately imagine there's an opportunity to basically triangulate very quickly on traits that matter for endpoints, targeting biology that is relevant and therapeutically actionable. So that's stuff that we'll do, but there's a larger community effort thinking about exactly these kinds of things too. Okay, let me shift gears to sort of a related point, and that is sort of taking advantage of not just one trait at a time, but multiple traits to try to, again, triangulate on what genes in the genome, regions of the genome are phenotypically interesting and worth spending my time thinking about. Okay, so I think about diabetes and heart disease a lot as if it isn't already obvious. Why think about that disease? Aside from it being a major worldwide health crisis, Coronary heart disease is a risk factor, is a comorbidity of diabetes. So diabetics are at twofold higher risk of getting heart disease or heart attack. So that's an intrinsically interesting thing, but why study genetics of those diseases? I would argue for two reasons: to study and understand biology, to get to the root causes of what these diseases are. Genetics is one tool in the arsenal to get at that. But ultimately, I'm interested in therapeutics, as I sort of implied. What I'd love, and I tell my graduate students this all the time, if you walk into my office and you said, Ben, I've got this region of the genome, it's associated with higher risk of diabetes, heart disease, blood pressure, adiposity, and circulating lipids. Can we work on this? I'd be like, yes. Let's stop everything we're doing. Let's go to the model systems biologist down the hall and start doing perturbations on relevant genes. Because ultimately what we'd like to do is find those sites, find therapeutic modulations that make sense, and kind of reverse the human genetic predictions based on the direction effects we could learn from humans. So how can we sort of collect the data and try to make better guesses about what sites in the genome matter for, say, type 2 diabetes and heart disease? So this is work that we recently had published in Nature Genetics, and it's really a very straightforward idea. So I'm trying to pave over a little bit of the math just to give you a little bit of the interpretation here. So conventionally, what happens in human genetic association studies is we collect a lot of data from individuals who have a disease like diabetes, compare their genetic variation to a bunch of people who don't have the disease. Large-scale consortium like Diagram or Cardiogram for heart disease have assembled large cohorts of exactly that. We compare genotypes at a specific site in the genome and ask, does the allele contribution at this particular site correlate with disease risk? And if we find a site in the genome where it's systematically different between cases and controls, exceeding the number of SNPs that we test in the genome, sort of like past this red line on this sort of like artificial normal distribution, we get really excited. So, oh my God, genome-wide significance. Can I have my Nature Genetics paper, please? Because I found 10 of them, and life goes on and everything is brilliant. So diabetes geneticists have done this. Heart disease geneticists have done this. A vector of human phenotypic investigators have done this. Typically, they do this one trait at a time. So diabetes genetics generally builds their GWAS. Coronary heart disease builds their GWAS separately from one another. So that seems a little bit artificial. We test the same SNPs across the genome as a whole, so why not actually just take those two distributions and combine them together? Okay, so if you do that, you get this sort of like multivariate normal distribution. Again, the particulars of the types of distribution that you're looking for are not really what I want to focus on, but what you'd really like to see is sites out here where the statistical support for diabetes or heart disease individually are not quite genome-wide significant. But if I told you that there was, in fact, a site in the genome that had this degree of evidence for both of these endpoints, it's probably itself genome-wide significant. Or put another way, we could probably construct this statistical distribution to model what this distribution looks like, and then thus look for sites that seem to have excess deviations on both of those traits. So I wouldn't be telling you this if it didn't turn out for this particular comparison that this set was completely empty. It turns out that this is a non-empty set. And let me point you to, in particular, this particular site. Now I'm tagging it with a gene named CCDC92. We don't have direct evidence, at least when the time we published this study, that this was the gene. I'll tell you a little bit about support on that in a minute. But this is a site tagged by this particular genetic variant. It's associated with type 2 diabetes at 10 to the minus 6, associated with CHD at 10 to the minus 7. The likelihood under our model that you'd see these two p-values itself, given the number of tests you performed, seems genome-wide significant. And the statistical signal that corresponds to heart disease and type 2 diabetes seems to map to the same genetic risk variant by statistical techniques that allow us to compare those association signals together. We can do that formally by conditional analysis. So here's sort of like the distribution of association for both T2D and heart disease. Here you've got the lead associated variant here, and if we condition the association on this lead variant, this association signal completely ablates for both type 2 diabetes and heart disease. So this data seems to suggest the model that there's a single statistical association that generates the evidence for type 2 diabetes and heart disease, okay? But if you were a skeptical reviewer, you might say, oh, well, you didn't show me genome-wide significance for an individual trait. How am I supposed to believe you? Okay, so let's get some replication data. For type 2 diabetes, we collected additional 20 or to 60,000 individuals, and in fact, this site does additionally associate in that replication with additional evidence, and if you combine that support, this now exceeds genome-wide significance for at least type 2 diabetes. So it suggests that the variant is in fact at least associated with diabetes and probably has some relationship to heart disease as well. What do we know about this region or gene? It turns out this variant is also associated with a couple of other traits, including HDL and waist-hip ratio. So it starts to indicate that this perturbation might be adiposity or related to fat depot or storage. This is sort of modestly supported by this observation that there's an EQTL in the region for adipose that's sort of modestly tagged by our signal, so perhaps gives us a clue at the gene, but probably need a little more support than that. And fortunately, this gene region actually turned out to be quite hot. In the last couple of years, a couple of investigators have sort of explored the relationship between genetic variation, insulin resistance, and adiposity, as well as heart disease. So for Sate Cathrasian and friends at the Broad actually identified another variant at this site in the gene that's a missense variant that's common that turns out to also associate with heart disease. Turns out this variant is actually not strongly linked to our variant. So again, it might be that there are multiple genes going on here, but the fact that the non-coding variant also associates with heart disease, you could imagine a simple hypothesis that loss of function at this gene might be heart disease and type 2 diabetes risk predisposing, i.e. it's the same genetic mechanism for the same gene, but we have to prove that formally. There's also some in vitro work that's been done where microRNA or interfering RNA knockdown of this gene, as well as a bunch of other genes, seems to imply insulin resistance defects when knocked down in adipose tissue. So they have a couple of different assays measuring lipid concentrations, adipose differentiation. You can kind of see the difference between control. There are less differentiated adipose cells relative to control in the CCDC92 knockdown. And the direction of effect seems consistent with a loss of function defect at this site. So again, it might be that CCDC92 is the right gene. It turns out this other gene is also seems to have the similar sort of phenotypic pattern. It's actually really close to this gene association, so it may be that there are multiple gene defects that actually are going on. But you can see that basically there's some story. It's got to involve adipose. It might involve one or more genes. There's definitely a heart disease and a type 2 diabetes mechanism in play. So if I had to spend my precious resources, of which I have very little, studying molecular biology of a specific region, I'm definitely going to put this on the list. Because if it turns out that this gene is easily knocked down, or that it's in a pathway that's knocked down, then maybe that leads to a type 2 diabetes and cardiovascular disease therapeutic angle. Once you start thinking about the data in this way, your eyes start to get a little bit bigger, and with the development of large consortium that collect lots of different phenotypes, you start to imagine doing additional comparisons that make sense, both on the cardiovascular, coronary heart disease axis, as well as the diabetes axis, just comparing pairwise each one of these traits to basically find things that may have not gotten to the genome-wide significant radar that fall just below the radar that seem quite compelling and give a very specific physiological or biological mechanism of action. Again, trying to find those things that you might want to place bets on and do functional experimental work on. Okay, so with the next couple of points, I want to give you a little bit of foray into our ideas about using machine learning techniques to try to, again, mine crucial sites in the genome, again, try to figure out if I had to spend my precious resources following up human genetics associations, which ones would I do, okay? So to do that, I'm going to have to give you just like the quick and dirty version overview of machine learning and its approach. So what is machine learning? It's the idea that you can give computers the ability to learn things without actually programming them. It's everywhere, and probably where it came, it spent a lot of time, and where I became more familiar with it is sort of image and video processing. But as many of us here might know, there's lots of enthusiasm from taking techniques and tools in machine learning to improve healthcare, to basically make a lot of money in finance, or maybe even one day when we have cars that drive themselves, we can actually make our computers a little bit easier and have Skynet drive us where we need to go. So it's everywhere. So how could this possibly, how are these ideas, could they be used in a genomics context, okay? But to do that, I have to invoke sort of like the canonical way in which one explains machine learning, and that is ubiquitously with the cat video. Okay, so if you're Google, let's say, imagine, and you wanted to know all the cat videos that were possibly available on the internet, okay, you could hire a bunch of people and have them watch videos and say, that's a cat video, and that's not a cat video, okay, but you don't have that time, you don't have that kind of money. Wouldn't it be great if you could teach a computer what makes a cat video, okay? So what might you do? Well, if you could only learn what sort of like the key archetypal features of what makes a cat, right? Like in this case, cats have sort of like fuzzy ears, sort of like a triangle nose with whiskers, and sort of like elongated eyes and so forth. If we could teach the computer, if the computer could learn that naturally from the data, then your kind of problem would be solved, because you can definitely show a computer, millions of cat videos, okay? So Google did this, and you can sort of see what the residuals of kind of what these models are. And I mean, if you stare at this enough, you can kind of see something that looks vaguely cat-like in this, right? Where the features, you see the eyes, the ears, the nose, and some of the whiskers here, okay? So once you have that model, then you can, of course, do what I say. You show it a lot of videos and make predictions and identify all the cat videos in the world, and I guess make a lot of money if you're interested and you're a cat aficionado, okay? All right, fine. So you might say, okay, Ben, why in God's name are you telling me all about these cat videos? I thought you said you were a human geneticist. Okay, so I would like to draw an analogy between sort of like seeing an image like a cat video and taking a snapshot of genomic sequence and its context. Sort of like a snapshot in time where there are sort of these features, some of which may matter for your phenotype of interest, others may not. But there's a ton of those features. So here's one, and you'd like to know which features matter. Right? Okay, so here's one of my favorite papers that came out a couple years ago from a graduate student in our Genomics and Computational Biology group at Penn. And this is a gene, LMO1, and it is a gene involved in neuroblastoma, so a cancer. It's one of these sort of really strong associations. It's like twofold higher increased risk of neuroblastoma relative to controls. So definitely a strong effect, probably the largest effect for this trait in the genome. Here's its sort of distribution of associations in space. You can see it kind of falls really near the LMO1 locus. LMO1 is a strong candidate gene, so the thought acting was sort of like, okay, this must be the gene, so let's do functional work. Okay, so then it's like, oh, well, how do we figure it out? This is a non-coding genomic region. There's not an obvious coding mutation that connects the variant to the gene of interest. Okay, but with a lot of resequencing and imputation, what you find is that there's this variant that escaped the original genotyping assay that you discover when you do targeted resequencing. It's quite a bit more associated than some of its lead variants, not in very strong LD, but it clearly harbors the majority of the statistical association evidence. And if you start to lay out functional genomics features in the region, what you find is basically this variant falls into a partially, somewhat of a conserved region, in a region of open chromatin, so accessible to transcription factors and other regulatory elements in the genome, and sort of hits enhancer-ish elements as well as overlaps the site of GATA3 ChIP-seq, so transcription factor occupancy genomically profiled. And then if you zoom in even more on this region, it turns out the variant itself actually physically disrupts a GATA transcription factor occupancy binding motif. So this gives you a really clear hypothesis about what the causal variant and gene and mechanism of action is. Because we know GATA is basically an enhancer transcription factor, so it probably somehow promotes the transcription of some target gene, presumably LMO1. So this was sort of like the lead figure of this paper, and the reason this paper is in Nature is because there's like a million functional experiments that sort of do the hard work to sort of prove that this is in fact the mechanism, that LMO1 is in fact the gene, and the biology here makes a lot of sense given what's found. Great. So if we could just find snapshots like this where we know exactly which functional elements to look at that allow us to identify and connect exactly the right variants, our job would be a lot easier than it is now. In this case, they zoomed in on a collection of, of genomic functional elements that make sense, but there are thousands of them in the genome. So it's a really sort of hard problem to understand which of the features make sense for a given trait. But if we could identify what those were, maybe we can get a hint of the causal mechanism involved. Okay, so let me try and draw this picture out in a little bit more detail, right? Okay, so conventionally what we have are a bunch of associated sites for a given trait. There are a bunch of variants that are associated, as well as variants nearby that are closely correlated with that variant. And we might get lucky and we find one particular variant that overlaps a bunch of occupancy or transcription factor sites that give us a clue that that variant may be the causal one and perhaps a mechanism that makes sense. In practice, what ends up happening is that in many cases, the GWAS leading association, the one that's the most strongly associated, doesn't really have any of this obvious functional elements attached to it, but it's something nearby in linkage disequilibrium that seems to have this sort of like compelling feature. This sort of looks like in the previous slide that I showed you, like what happened at the LML1 story. So the association data itself can't tell you which one of these is true, but if we have this sort of layer of functional information, that might give us that clue that we're looking for. Okay. We're not the first to sort of try to think about this kind of problem, trying to deduce causal variance from genomic data. There are a range of tools that think about this problem, but they all have one property in common, which is to say that they're generalist. They don't care about the phenotype that you're looking at specifically. They're learning their model, they're training their model based on the entirety of a catalog of human genetic association data. They're training their models versus simulations, which perturb every variant in the genome. They're taking large amounts of publicly available data that may not even be phenotypically specific or relevant to your trait of interest. So they definitely work, but don't necessarily work for the phenotype you're interested in. So for example, if I thought that I was studying immune-related disease, I'd probably be more inclined a priori to look at functional genomic annotations that were relevant for immune cell types. That kind of makes sense, and there is a collection of supporting data that supports the hypothesis that gene regulatory features map to the phenotype in tissues that are relevant, but there's no sort of like computational analog to sort of connect specific tissue relevant or phenotypic relevant information to the predictions that we're making on a particular causal locus or variant. Okay, so what we'd like to try to do is like take that snapshot. So basically what we've got from our human genome-wide association data are a couple of cat videos. We've got like these snapshots of things that we know for sure relate to diabetes or we know for sure relate to heart disease. And we'd really like to understand what is it about that site that makes that a type 2 diabetes locus. We'd like to know the transcription factors, the sites of open chromatin, and in what tissues sort of are like most enriched in these sites and sort of give us the best chance of predicting what these genomic sites are. So if we have enough of these in the data and we have enough negative controls we can match to, perhaps we can build a model that actually distinguishes between these two cases. So that's what we're proposing. So what we can do is take, to build the snapshot of the features, we'll take advantage of publicly available data like ENCODE and the epigenomics roadmap. We'll take advantage of established associations. So our cat videos are our established sites for type 2 diabetes. We'll apply them to a simple machine learning model, some regression model. We'll generate scores and generate predictions about which variants and which features we think matter for predicting which sites we think are relevant for type 2 diabetes and heart disease. Okay, so let me walk you through sort of our work on type 2 diabetes, which is immediately a tough problem, but here's what we did. We know of 100 sites in the genome that are reproducibly associated. We can match those to sites in the genome that we know for sure are not associated with diabetes because we can measure their association and they don't seem to relate. We can also match for a couple of other ancillary features like distance to gene, the allele frequency of those sites, things that we don't really care about. We care more about what functional annotations matter. We pick out epigenome roadmap and ENCODE as well as species conservation. In addition to that, we actually took the painful step of going through of literature curation and finding additional epigenomic features that map to open chromatin or transcription factor occupancy, specifically thinking about tissues that we think might be relevant for diabetes. So those include pancreas, adipose, liver, muscle, intestinal cells, so gut, things that we think might matter. So with that, we can add an additional couple of hundred features that we think could be relevant for diabetes. And so what we'll do is we'll present all those things to a machine learner and have it tell us what it thinks the features are and try and use those features to distinguish between associated variants and non-associated variants. I won't belabor this slide too much, but for the machine learning aficionados in the audience, the model we're using is Lasso. That's implemented in R. I'm going to show you the holdouts, not the cross-validation, but take my word for it, the cross-validation works reasonably well. Maybe our model's a little bit overfit, but that's why we're relying on generalized holdouts to show you our prediction accuracy. So how are we measuring how well we're doing is really based on this holdout set. So I told you that we had 105 sites that we're using to build and train our model, But it turns out whilst we were doing that, the pursuit of identifying genome-wide associated loci for type 2 diabetes continued, and there were additional 28 that were discovered that we didn't use in our training set. So that gives us a way of really just assessing how generalized our model is. If it had no prediction accuracy, and our model was horribly overfit, our model would not do well at predicting these sites at all. So we can assess that very directly. Okay, so I'm going to show you a couple slides, and in case you're not familiar with sort of ROC curves, here's how they work. What we like are cases like this. I'm going to show you sort of like the true positive rate-ish over the false positive rate. A great predictor would look like this. In practice, we'll have stuff that's sort of less well predicted, so accuracy are much less than that. So here's what it looks like for our models and for competitor models that we are available that are conventional. Sort of like the best predictor model that we have out there is this Guava tool. It in fact uses epigenomic features as well, so it's probably not surprising that it does better than some of these comparator methods. But our model that includes all of the features that it learned from Epigenome Roadmap, as well as the ones we discovered, even does better than this. So you might say, Ben, I'm really not that impressed. You know, this is like a 10% prediction accuracy. You know, you told me that you want to make no error, you know, make all perfect predictions and no mistakes. Well, it turns out this is a super hard problem, and some of the better machine learning prediction tools that are out there are high profile, and they make prediction accuracies basically on the order of this or actually less. So we would argue that on a model that we haven't optimized, on a collection of features that we don't think fully capture all of the complex etiology of diabetes, the fact that we're doing better in a holdout prediction set relative to this other method is actually probably in advance. And this is one type of machine learner. Again, for the machine learning aficionados in the audience, there are a range of different types of learner that are even more flexible and dynamic than Lasso is. So actually a lot of room to improve the prediction power, I think, of this particular approach. One thing that's nice about Lasso, though, is it gives us a little bit of confidence about interpretation. So there are a range of different features here that are selected that are really quite nicely relevant for what we thought mattered for type 2 diabetes. We're selecting features like the pancreas, adipose, and liver for both transcription factors that we know are relevant for the disease, as well as transcription factors that we might have thought were relevant, but maybe have not had considered different types of tissues that were connected. And remember, we gave the model, we didn't tell the model that these were the features it should have selected. It told us what these features were. We gave it like the entirety of epigenome and ENCODE. So that's like a vector of a huge number of cell types that have nothing to do with diabetes, profiled for a range of different types of traits. Molecular markers that may or may not be relevant for type 2 diabetes. So while it's a little bit of an art form and one has to interpret this a little bit cautiously as one does when thinking about features selected for machine learning models, we take this as a cautiously optimistic sign that we're picking things that we think are disease relevant and matter. So let me tell you about one or two examples that sort of give you confidence that we're picking out things that make sense. So here's one locus that we think makes a lot of sense, PDX1. It turns out PDX1 is one of these core genes that are involved in the development of pancreas. We know that coding mutations in PDX1 cause monogenic forms of diabetes. We didn't actually add any, when we were doing the genome-wide prediction, this particular gene wasn't actually added in our training set. So this belongs to a set of the genome we're now trying to predict and find interesting sites to follow up on. We didn't train on this model, but our model told us that this particular site was hugely relevant for diabetes. So here's one, the leading association, the most associated site in this region is sort of in this dotted red line. But actually, there's this site over here that actually overlaps a lot of marks in pancreas. Again, because we think PDX1's action is relevant in pancreas, the fact that there are transcription factor occupancy sites as well as marks of open chromatin that seem to overlap this one variant that's in strong linkage with the most associated variant in this region suggests to us that A, this region is probably relevant, and B, if we had to pick a causal variant in the region, we'd probably look very carefully at this site rather than this other site that doesn't seem to have these overlapping markers. Here's another example of another gene. Again, this is not one that is established for association for type 2 diabetes just yet, but it kind of has that LML1-like property, where basically here's the upstream promoter region of this site. The most associated site here is in this red dotted line, sort of far away from the promoter site. But there are variants that are really strongly linked to this site that basically overlap marks that our model would have predicted are highly relevant for diabetes, including pancreas, liver, adipose, et cetera. So it suggests that somehow there's this functional regulatory element here, perhaps, that maybe alters regulation or somehow in a way we don't understand with this transcription factor that might relate to diabetes. So you might say, oh, well, that's a stretch. There is some model systems data that has knocked this gene down, and mice actually have some glucose-related defects when this gene is knocked down. So again, some small amount of convergence with human genetics and model systems with this particular mark. But this is kind of the thing. If I had to spend my time thinking about things to study, I'd probably pick sites like this. Okay, to give you one other proof of concept example, let me tell you a little story about platelet biology. So again, if you don't know anything about platelet biology, it turns out that what makes platelets, so the particular cell types that are involved in coagulation and hemostasis in blood, involves differentiation or megakaryopoiesis. And it turns out that we are only starting to learn a little bit about this process. We can kind of make platelets, but we don't know all the biology around them. But that's hugely important because we don't have enough platelets. If we had more of them, we could definitely do much more clinically directed innovation and implementation using that resource. So if we could just figure out ways to make more of them, it would be really helpful. It turns out we do know a little bit about this process. So it turns out there's this core transcription factor complex that is involved in the job of regulating genes that are involved in megakaryopoiesis. We know some of those players. Those include GATA as well as some of these other genes. But it turns out we don't know everything. So we know this complex is involved. We don't know all the players in this complex, and we definitely don't know all the places in the genome where this complex regulates genes. So if we could just figure out ways to build that network, we could probably understand more about this biological process. So if you can guess what comes next, we'll play our machine learning game. So it turns out we know of a number of sites in the genome that reproducibly associate with platelet traits like platelet volume or platelet size. We can compare those to negative controls, collect a large number of genomic features that we think matter for chromatin, just as we did in the diabetes case, apply our machine learning engine, develop a score, and then find places in the genome we think look like megakaryopoiesis or platelet-related trait sites in the genome. So we build our score. What are the features that are selected? Again, comfortingly, there are features that sort of make sense that we would have thought if we were biologists trying to pick the right features, we would have guessed at. So these are basically involved in the complex directly. They are enhancer element marks in sort of cell types that we think are closely related to the phenotype of interest, and a couple of other marks that seem a little bit more distantly related, but somehow involve enhancer activity. So it suggests that our model is picking out something that makes sense and perhaps indicates regulatory biology might be the thing, enhancer activity might be a typifying feature of what makes a megakaryopoiesis platelet-related trait genome-wide locus itself. So we can, again, as a positive control, ask the question, how does our score correlate with sites of established association versus not? So not surprisingly, it does. The higher the score sort of typifies genome-wide associated loci. It turns out that this is true not just for sites we know are established, i.e. those sites we use to train the model, but also correlates strongly with sites that have not yet reproducibly associated. So the stronger the association from humans, the stronger the score. When you go to the scores in the rest of the genome and ask, for the nearest gene, given the caveat that the nearest gene might not be the causal gene, what are the annotations functionally that are attached to that gene? Encouragingly, those functional attachment relate to blood cell-related ontologies as well as megakaryocyte differentiation. And again, these are SNPs that weren't used to train the model. So this is orthogonal biological support for the idea that the score maps to megakaryocyte-related biology. It turns out that the score correlates with genes, either the transcription factor element upstream or the coding sequence itself. Again, we didn't tell the model that, but that's sort of a non-random association that's consistent with this idea that the non-coding variants seem to somehow disrupt regulation of genes that are involved in megakaryopoiesis. So from the established sites, we can look at them carefully, and we can probably guess at or make a case for one or a handful of causal variants at perhaps like 80 to 90% of those sites. And that's way better than what we were doing previously. But that's still way too many for us to actually take to the bench and prove functionally, okay? So remember, I want to figure out ways in which we can go to like the 1 or 2 sites that we have a very clear biological mechanistic hypothesis that we can test, because we can really only test 1 or 2 things. So really, how should we actually prioritize those things, okay? So then we go back to our biological first principles, okay? So we know this complex is involved. So let's go to sites where our score makes a strong prediction, where there's a, 1 or more of those variants are tightly linked to SNPs that score highly, and that there is a transcription factor that is directly disrupted by one of those variants of interest. So again, like GATA. So this would give us a really clear biological hypothesis we could go and test in the bench if such a site existed. And I wouldn't be telling you this if it didn't turn out to be the case that there were at least 1 or 2 of those such sites. So the first one, again, is a story that was already known and was already established. So there's this site here where our score is pretty potent. There are linked sites to this site that are also pretty well associated. It's near a gene called PIK3CG. This gene seems to somehow be biologically related to megakaryopoiesis. If you now line up all the functional predictions, functional marks that our model predicts, you can see that there's this nice overlap between enhancer functional activity marks, as well as it, of course, by definition, disrupting a GATA binding site. It turns out that this paper, noted almost all of this except for the specific marks here, but they also know that this is an EQTL for this particular gene, for this particular variant. So the variant that seems the most likely to be causal based on this functional genomics profile is also an EQTL and sort of co-localizes that EQTL signal. So it gives you like a very clear mechanistic hypothesis. Disruption of this somehow disrupts platelet function in some way and gives you a clear hypothesis to test. We would go and test that hypothesis, except that we already know everything. So, okay, can we find another example of something we didn't know that also has a functionally compelling signal? So here's one other example of just that. So here we've got the GWAS-associated SNP, not particularly strongly scored, but it turns out to be strongly linked to a variant that we would have predicted from our model is strongly scored. We stack up the functional genomics evidence. It seems like there is perhaps a GATA binding site here that's not one of its sort of canonical disrupted bases, but could be, could disrupt a GATA occupancy site. It's a little hard to see here, but there is a small amount of evidence for GATA occupancy at this particular site. So again, gives us at least a functional hypothesis to test. Maybe it's this gene, maybe it's this GATA occupancy site, maybe that disrupts platelet site biology. And the nice thing about studying blood cell related traits is that you can actually do that, right? Like unlike a diabetes experiment, I don't necessarily know what the cell type of action is, but for blood cell related traits, we can actually take megakaryocytes, try and differentiate them through some genomic editing experiments to try and understand whether or not this is actually the causal gene, and if so, then actually go about disrupting the variant to see if actually the variant that we would have predicted was functional is actually the one that relates to the trait. So hopefully that gives you, there's a bunch of other things to do, but hopefully that gives you a clue that the information we've already gained from genome-wide association studies is telling us something about biology, and that machine learning tools, at least in our hands, give us a way of sort of sifting out things that we think are relevant and interesting, gives us clues of things that we can follow up. Okay, so for the last 1 or 2 minutes, let me just hit the last point, which is sort of like, okay, well, what do we do now, okay? So it just turns out that human genomics, much to my chagrin now that I'm a tenured associate professor, moves incredibly fast, which means that there are a lot of people, really smart people, doing a lot of really cutting-edge, blazing work, collecting a lot of data. So it bears mentioning that probably we should take a little bit of pause to think about what that means for what experiments we could do, or at least where we're going, okay? So one thing that I think is interesting is that the GWAS of tomorrow is basically here, okay? So when I was growing up, we were really excited to pull together thousands of individuals who have diabetes or heart disease and compare them to controls. And those bygone days are over. We live in a biobank era now where whether or not it's the UK Biobank, Geisinger, the Million Veterans Project, just these tons of resources where today it's 10,000, tomorrow it's gonna be 100,000, in 2 or 3 years it's gonna be millions of people, where you've got genetic data already available, you've got those data attached to electronic health records, so you have a great opportunity to connect genotype with phenotype if you can handle the QC and the technical details of that data well enough. And that just blows my mind. So it's just like, okay, like, we got to think really carefully. Like, we're going to make a lot of discoveries when all this data gets collected, right? So we've got to think about what the particular loci that are interesting that we're going to spend our time working on. If we believe in the omnigenic model where there's like everything's associated, we're going to find thousands of things that are relevant, but we're not going to want to study all 1,000 things. We still have to be clever about what things that we actually want to discover from, follow up on all of these activities. One other thing that I also think about a little bit is sort of like from, in a parallel, closely related quantum reality, there is the GTEx project, which is this large-scale effort to connect genetic variation to change in gene expression for all genes in the genome. And they're like pioneers, even here at the New York Genome Center, that think about this particular set of problems a lot. And I'm just a passive bystander to this. I'm a utilizer of data, not a data generator. But from the papers that are coming out, and we can sort of have this discussion offline too, it seems to me that there are a couple things that like GTEx has taught us aside from all the basic discoveries that have been made, which is to say that a couple things. A single genetic variant can indeed change the expression of multiple genes in multiple tissues. So it's not always simple. It is more often complicated than it is simple. Not only that, it's sort of the obvious thing, which is it's not just one variant that changes the gene expression of that gene or genes in multiple tissues. There are multiple variants that do the same thing. So that to me makes me think hard about what that means for sort of like traits that aren't human, not gene expression traits, but like human phenotypes. I don't think that it's unreasonable to suggest that like type 2 diabetes or heart disease or height or schizophrenia is going to somehow be different, radically different from this really basic model. So it's probably the case that actually many of these sites are really, really complicated. So if that's really true, I think that means we should think hard about how we tackle this mapping problem a bit more effectively. Like, do we need to rethink the model that a single variant impacts a single gene with a single genetic mechanism in a single tissue? Or is it actually more complicated than that? And if it's true, we probably need to think about improvements to how we approach the mapping problem and the functional follow-up problem. This is another paper that sort of just makes me sort of cringe a little bit, but also I think makes sense, and it's a really intuitive one, right? Okay, so we think a lot about, I think a lot about complex disease, and the canonical paradigm when I was growing up and went to graduate school was there was Mendelian disease, it's a single gene or a few genes, and that's different from complex disease, which is many genes or polygenes, so hundreds of genes in the genome that matter for that particular disease. So this comes crashing down with sort of this very nice summary paper that kind of pulls together a bunch of different evidence from a variety of different sources that makes the case that it's not polygenes, it's not just a finite set of a few genes, it's probably actually almost every gene, so omnigenic. So that in fact, there are core pathways that are sort of connected in peripheral ways through changes in gene expression. And that's sort of like what explains why phenotypes are as complicated as they are, or maybe one reason why. So when I read this paper, I think, okay, yeah, I mean, what are the core pathways then? Like, we've got to think hard about what gets to these stars, because I don't want to spend my time studying peripheral genes. I want to study these genes, okay? So that sort of brings to this front, like, how do we identify those from the data? I also think a bit about like therapeutics, right? So like drug development works, okay? So before we knew or thought about omnigenetics, we like tried to, we went to mouse and we knocked genes down and we found things that protected mice, and then we developed small molecules for those protections, and we developed drugs based on those things, and those drugs definitely help people not have disease, okay? So that works, but like, what is the process by which, in this context now, this development process has actually worked? I think it's worth thinking about. If it also turns out that everything is involved, right, okay, in the context of not just one phenotype, but like lots of phenotypes, okay, it seems to me that we need to think about phenotype min-maxing, right? So I want a core pathway that perhaps is a really strong perturbation on the disease I'm primarily interested in alleviating, that sort of like minimizes all the things that I think are going to give me a bad time, like is going to, not disrupt too much my cardiovascular disease risk, definitely isn't going to give me schizophrenia, blah, blah, blah. So thinking about, like, even if we identify a core network, not all core networks are probably going to be therapeutically actionable. Probably a subset of them that sort of like, we need to find the optimizing functions that tell us which core pathways matter, not just for the disease of interest, but also don't disrupt too much peripheral other phenotypes, or at least if they do, it's something we can manage and deal with. Okay, so with that, let me just summarize. Hopefully what I've convinced you again of is this key idea that computational and statistical approaches using human genetics data can help pinpoint biology that's interesting to study. I told you about tricks with causal inference that can be used, phenotypic mapping methods that sort of triangulate at not just one but multiple traits, that you can sort of optimize locus and causal variant discovery and follow-up using machine learning tricks, that works for type 2 diabetes and platelets, and we're going to prove that functionally, hopefully. And that we've learned a lot actually from this sort of endeavor of collecting a lot of big data from human genomics, and we still have a lot to learn from those. But I still think it means we need to choose wisely about what we want to follow up and how we interpret that data. So thanks for your attention. This work would not be possible without a great lab and a sea of collaborators that have helped out a lot. And I'm happy to take questions.
You mentioned that you're focusing your work with the idea of developing therapeutics. You also mentioned that you focus on one disease pretty much at a time. At the beginning, you talked about PCSK9, which lowers LDL cholesterol. But it seems to do that at the expense of creating fatty liver and even NASH and hepatocellular carcinoma. The other question is, are you making hidden assumptions about the heterogeneity of mechanisms in the populations that you're studying? Just to pick that example, an APOB mutation will also lead to failure to transport VLDL and very low LDL cholesterol, low triglycerides. Different gene, somewhat different mechanism, and suppose you had a lot of heterogeneity underlying type 2 diabetes. In other words, the surface of the illness, signs and symptoms, which is the clinical bedrock on which everything is based, may really be, things may be much worse than you think. I mean, do you have any way from your statistics of dealing with that?
Yeah, very relevant question. So the NASH and PCSK9, yeah, I think this is one of the tricks and challenges with trying to do these sorts of causal inference techniques, right? Look, what you'd really want are variants that only relate to that trait and nothing else, especially nothing that's complicated. So if it turns out that loss of function causes fatty liver, that's probably going to be a bad time from the point of view of developing a therapeutic around it. Nonetheless, Regeneron and others are still trying to pursue that to see what's going to happen ultimately. But like in the idealized case, you definitely want to try to, I could imagine a world where with very rich phenotypic information, we might not only be able to find those perturbations that matter, but then expected complications that might have also arisen from specific gene perturbations. And if we had that, maybe we could optimize those things. In terms of the second question, which is what happens when there's a lot of genetic heterogeneity, how do you make these sort of connections? And I mean, I will concede, like, diabetes is definitely quite heterogeneous. There's no question that the disorder is a multi-system disorder with multiple types of different physiological mechanisms. And there are two ways in which that manifests. Like, there's like the statistical one, which I think about a lot, which is what we'd really like to be able to do is educate our models to make predictions which try to understand what that heterogeneity is. And so if we can measure that with functional annotations, the right cell type models, educate that with other phenotypes. I talked about just like a model trained with diabetes loci, but there are BMI-associated loci, fatty liver, adiposity. We could use those other type of phenotypic measures to try to get a little bit at that heterogeneity and then build our models a little bit more intelligently. So that's definitely something to do. With respect to like the clinical impact that I think you're getting at, which is like, do we worry much about causal inference in this context? I think this is sort of what you're saying when there's a lot of heterogeneity. Oh yeah, almost certainly. The methods definitely make assumptions, and we should be mindful of those things. So for example, in the case where we do this one variant at a time or a specific variant at a time, we definitely assume that the distribution of variation at other sites is sort of random with respect to that. So there, carriage of loss of function at this particular site doesn't determine whether or not you carry additional variation at some other site in the genome. If that assumption was not true, all of our inference would go off to naught. At least from what we understand from meiosis and other things, we tend to see, we don't think this actually happens in practice, but we have to be mindful of the assumptions we make for sure.
If you're going to spend $1.8 billion bringing a new molecular entity to the market, and you've got lots of different mechanisms, and you want to target just the mechanism to minimize side effects, it seems to me you're going to need an enormous amount of drugs and money, unless there's some new paradigm.
Yeah, that's definitely a possibility for sure.
Sorry, I have a question about the machine learning aspect. You mentioned the lasso technique, and I don't know how old that technique is, but is it like 10 years old at least?
Oh, it's amongst some of the first to think about this.
So is the real bottleneck in the work you're doing, and I was very interested in the mapping part of it in terms of like looking at the data, is the real bottleneck that you need faster computers and more advanced machine learning techniques, or is the real bottleneck the data and the mapping?
It's the data. I think if we'd be much happier if we had a truth, like if you handed me 1,000 cat videos, I'd be much happier. We might be able to do better with different machine learning techniques, but I don't think at least with the vector of features that we have here, I don't yet believe it's a computationally intractable problem, or not something we can't solve with like 1,000 nodes for a couple days, like something we can solve. So I don't think it's that challenging. But you know, the future is bright. We're doing a very good job of collecting a large amount of functional genomics data. Like I mean, I don't know how you all feel here, but like I can barely keep up with tomorrow, like Nature publishing some high throughput sequencing technique that's like CRISPR solve it all, right? That just generates some other annotation that we never thought about that matters, and that we now collect at high throughput. So, you know, in the long term, yeah, I'm not sure how it looks, but for now, I don't think that's a problem.
So 105 positive labels is quite limited for any supervised learning technique. Is there any unsupervised technique you have tried?
No, but I literally have a rotation student this term who I pitched this to, and she is excited yet timid. She has to get situated in her classwork, but that's basically the next set of experiments we're going to do, is basically try a different panel of learners to see if we can optimize this a little bit better. I would be much happier trading a little bit of interpretability, or maybe a lot of interpretability, for improvements in holdout accuracy. I'd be happy to make that trade. So
I was hoping for just a few more details on one of the slides that you gave as to how you chose these features to feed into GLM-net. You showed various organs. I think you read 442 papers, something like this. Could you give a bit more details as to concretely how that process went?
Yeah. I went through that pretty quickly. It's actually really straightforward. It's two phases. One is there are two large-scale, sort of phenotypically unbiased functional genomics resources, the Epigenome Roadmap and ENCODE. They have functionally profiled transcription factor occupancy, histone modifications, et cetera, and they have like thousands of these things profiled in a bunch of different cell types, as well as primary tissue. So step one, take that like 3,000 feature database. Then step two, tell your postdoc to read a lot of papers and find data sets and convince the PIs to release their data. And so that comprised basically like 44 papers and an additional 442 features. And there it was literature mining. So basically what we were looking for was to augment what was already publicly available from ENCODE and Epigenome Roadmap. So we were diabetes minded, so we were thinking, okay, what tissues are relevant? We have a sort of a short list of prior tissues that we think are interesting. Okay, what kind of marks are we interested? Well, we'd love transcription factor occupancy, but we'd basically take anything almost. So you'd think that it'd be harder, or that was like cherry picking and bias, but it was literally like us trying to get to Geo and find anything we thought was relevant and pull out as much as we could.
So step one actually doesn't exist from what you're saying, right? ENCODE exists, and then step two was we read lots of papers. Okay, so kidneys are important, pancreas, and so forth. I'll go and I'll download the data for pancreatic pancreas and kidney and so forth, we'll feed that into GLM-net, and those are our features, right?
Yes. Yes,
Okay.
and so what goes into GLM-net is basically like 3,000 plus 442 things, and then
Right.
it selects a subset of those.
I
Yeah.
see.